So let me just, before I start the presentation, present myself. So we shall share some information. So I'm an Israeli physician and scientist. So I graduated the medical school in Israel, in Jerusalem, in the Hebrew University. I then was trained as a surgeon, but quite deep into my training as a surgeon, I somehow decided that medicine is maybe not for me. And the reason was the following. If you are working in a hospital, and I worked for quite a while in the hospital, many of the patients are coming when it's too late. For example, if you are thinking about a patient with Alzheimer's disease or Parkinson's disease, they are showing up when already 80% or more even of the brain function that is related to what they do is already lost. So, you know, in Parkinson's disease, for example, the patients are losing stability. They cannot write because they are trembling, they are shivering, they cannot button their shirt, they cannot dry after the shower, they cannot cut the food with a knife and hold the pieces with a fork. And the region in the brain that is responsible for our stability is called the substantia nigra. I shall try not to use any technological terms, but the substantia nigra has to degenerate by more than 80% when the patient is showing only the first symptom. So the patients are coming when the process is already very deep. You know, from time to time, we are hearing about a patient that, you know, was a jogger or a football player, and all of a sudden he or she succumbed, you know, falling dead while playing or while running, while jogging. And people say, wow, he was healthy until it happened. No, he was not healthy. You know, the American military made autopsies, post-mortem operation on soldiers that were killed during the Korean War. And already at the age of 18 or 19, they found that the arteries, the vessels that bring blood to the heart itself are already starting to get occluded, to get kind of rusty. And then it takes decades until they are completely occluded. And then once the very last little lumen get occluded and there is no blood flow to the heart, then they get what we call heart attack and they fall dead. So the disease actually was incubated in them for years before they basically either, you know, got heart attack and somehow they managed to survive it because they were, you know, taken to the hospital on time, or if they just fell dead. And the same for cancer. You know, when people are complaining about cancer and they come to the hospital, well, they don't complain about cancer. They have some other complaint, and then the physicians in the hospital discover that they have cancer, and then the tumor is already very big, and in many cases it's a little too late. Not a little bit, but in some deadly cancer, it's so. I had this naive idea that if instead of being a physician and seeing patients that it's very hard to help them, I will rather go to science. And in science, I may be able to discover the root of the diseases, why people get sick at the very beginning, how diseases start. I must admit that at that time, it was a very naive thinking. In retrospect, it turned out to be true, but I thought that it was a very daring type of a dream to think. So I decided to go to science and to do a PhD. And with thinking that I will be able later on to better understand the mechanism of the initiation of diseases, why diseases start at all. As I said, it was a very naive thinking at that time, but it turned out later on, after more than 3 or 4 decades, that I was right in my feeling. So I enrolled into a PhD program in the university that I'm currently a member of, in the Faculty of Medicine of the Technion, which is Israel's premier technological university. And I started my PhD on protein degradation. So let me share the screen now, and then I will. Let's talk about what I did. I'll explain it to you in a very, very simple language, and then I'll expand a little bit to modern medicine, so you'll have a better view. But you'll see how we started the story. So the story is about how proteins are destroyed in our body. So what are proteins? Proteins are basically in every one of our food. Let it be vegetables, let it be dairy, let it be meat, let it be fish, let it be fruits. So they are proteins, but we are not using them directly. Why we are not using them directly? Because if they will be injected directly into our system, they will elicit an immune response. You know, the fish has its own protein, the cow, its own protein, the chicken, its own protein. So what we are doing when we are eating all this wonderful food, we are digesting these proteins into their very basic building blocks, which are called amino acids. We are then digesting, we are absorbing from our intestine the amino acids, and we are synthesizing our own proteins. Now, why do we need proteins? What are proteins? So let's just look at this cartoon. You know, proteins are helping us to digest the food. They are enzymes. You can see it, you know, on the left upper corner, you know, the digestive tract. Proteins are making our antibodies. On the second from the left, you know, we are now, probably most of you are, or all of you are immunized against coronavirus, against influenza, against other diseases. The antibodies that are immunizing us against diseases, either bacterial diseases or viral diseases, are antibodies. Proteins are making our muscles. Look at this weightlifter. You know, the muscles, the power of the muscle is derived from two proteins that are sliding against one another like cogwheel. These proteins are called actin and myosin, and the force is, you know, made. You know, we are able to exercise force and to lift weights by the movement of proteins. If we look at the lower part of the cartoon, you see the bones. So what gives us stability, you know, the support to the body, the bones are made of proteins. If we look at the very right lower panel, you know, the blood, you know, the red blood, the color is coming from a protein called hemoglobin. And what hemoglobin is doing, you probably know. Hemoglobin carries oxygen from the lungs to the tissues. Oxygen is our fuel, basically, and takes back from the tissues carbon dioxide that we exhale in our air. So we take in oxygen and we exhale carbon dioxide. So the carrier of these gases is a protein called hemoglobin, and hemoglobin has an iron molecule, iron ion in it, which gives it the red color, and hence our blood is red. So you see, you know, the eyes and the ears and the tongue that can taste and see and hear, you know, the hearing sensors, the receptors, the seeing receptor, the vision receptors, and the taste receptors are all proteins, and they transmit the signal. And then you can say whether the food you are eating is tasty, or whether, you know, it's rotten, whether it's sweet, or whether it's salty, whether it's bitter. You know, we can see colors, we can see shapes, we can identify them in our brain. All these activities of the body are mediated by proteins. So the body is made of about 25,000 different proteins, 25,000 different proteins. And for me, if I want to put it in a metaphor, they are playing a wonderful symphony. And the symphony is called the Symphony of Life. And amazingly, this symphony orchestra doesn't need a conductor. Each protein knows what to do and when to enter into the symphony, when to exit from the symphony, and they don't need a conductor. Now, the issue with the proteins is that they are very unstable. But I'll give you a little bit of how they look like. Basically, the proteins look like words in the language. Let's take the English language or the Hebrew language, doesn't matter which language. An average word is made of 6, 8 different colors. Let's take the word mother. So we spell it M-O-T-H-E-R. So this is the right spelling. But if we make a mistake in the spelling, let's say that we are now spelling mother M-A-T-H-E-R, so it's a spelling mistake. If in the protein there is a mistake, and we'll talk in a minute about what mistake, it's called a mutation. There is a mistake in a protein, and the protein doesn't function properly. So what are the letters of the proteins? The letters of the proteins are called amino acids. Look at the upper right panel. You see the amino acids are, connected to one another in a protein, like in a chain. So one amino acid, the blue one on the right side is connected to the red one, to the green one, to the yellow one, and then to a red one and another blue one, and so on. And they make a word that is called protein. The main difference between words in the language and words in the protein language is that the length of the words in biology is much, much longer. An average protein in the human body is made of about 500 letters, 500 amino acids that are connected to one another. Now, for us, because people made languages, the letters are written in a very linear way. So the eye can see them and read them in one shot. Proteins are not like that. They are folded in the three-dimensional space. And you can see that they are making helices. You can see on the right side, on the second from the top figure, you can see that they are making helices. So they are kind of like spring. They are making rings, and they are making sheets. If you look at the left side, you see that they are making helices, they are making sheets, and they are having a very complex structure. So you can see on the left side, on the upper top figure, you can see how we are taking the chain, and we are starting to condense it into a helix and into a sheet. And if you look at on the left side, but on the right lower panel, you can see the entire structure of the protein. This is an Achilles heel in the protein structure because they are very sensitive. They are sensitive to irradiation. They are sensitive to mutation, because if there is a mistake in one of the letters, they don't fold properly. And if they don't fold properly, they are not functional anymore. They lose their function. They are also very sensitive to heat. Think about it. Think about this simple experiment. Everybody can fry an egg, right? You take an egg, you break it on the rim of the pan, and you throw it into the boiling oil, and in 2 minutes you have a fried egg. But nobody in the world can take the fried egg and put it back into the shell. Not because the shell is broken, because you cannot reverse the changes that the heat imposed on these proteins. The changes are irreversible. If you take, for example, a fresh egg and you put it into boiling water, you don't even break the shell, you get a hard egg. Nobody can take the hard egg and bring it back to its native form. The heat denatures protein. It's irreversible. So you see that proteins are very sensitive to heat. You might argue that I exaggerated because I put the proteins in a very high temperature, in the temperature of boiling water, of boiling oil. But let's do another experiment. Let's take cheese or sausage and put it at room temperature. We are not put it into any excessive heat and see what happens. Fungi start to grow on them and they got rotten. So, if you leave a piece of fresh meat or milk or cheese on the table for a few hours, for a day, let's say that your mom sends you to the supermarket, you go, you bring it back home, you put the milk or the cheese, instead of putting them in the refrigerator, you put them on the table, you come back home after a few hours or after a day, it's gone. Why? Because even the room temperature is deleterious to proteins. They get denatured, misfolded, rotten, and non-functional. Now think about the human body. The human body does not live in room temperature. The human body is living in a much higher temperature. We are living in 37 centigrade, exactly. Think about it. We are living in 37 degrees. Now, 37 degrees, as I told you, and as you already know, is deleterious to proteins. So what is the difference between the meat that we left on the table to the meat that is in our muscle or in our blood? Basically, there is no difference at all. The same meat that is in our muscles is the same meat that was taken from the cow, from the chicken, from the fish, and was left by mistake on the table. Except that our meat in the muscle, in the blood, in the stomach, in the eyes, in the tongue, in the ears, in the skin, is in a living body, while the meat on the table is coming from a dead body. Now, living organisms have an advantage. We have a system that can take proteins that are getting damaged, let it be by temperature, let it be by irradiation, let it be by oxygen, let it be by air pollution that is made by cars, by the heavy industry. We can take these proteins that are getting damaged all the time as I speak to you now, and as you sit in the hall, your proteins are getting damaged non-stop online all the time. But unlike the proteins on the table, our proteins are in the living body, and the living body has a system that destroys the bad protein and makes new one that replaces them instead. And what we discovered when I was a graduate student is the system that degrades rotten, misfolded, denatured, harmful, non-functional proteins. And you see here, proteins in the body that are mistaken are making a lot of trouble. Look at mutations. Well, this is kind of an imaginary drawing of the person who did it, like the fish that had legs like a chicken, or the dogs with the 3 heads. But, any mistake in the letters that in the DNA that codes for the protein, any mistake, you can see here the DNA on the right side, you can see that there is, a mistake, a mutant protein, the red was put instead of the black letter in the DNA, can cause a very harmful mutation. As a result, a non-functional protein will come out, and this protein may be harmful. So what we discovered? We discovered a system that destroys them, and the system takes the protein to be degraded. You can see it on the left side, this kind of a brown snake, and attaches to it another protein. It marks it for death. Actually, the Nobel Committee called this mechanism, or the small protein, the blue protein, the ubiquitin. It's called ubiquitin, the small protein that attaches to the bad protein. It called it the kiss of death. What the ubiquitin is doing, ubiquitin is like a glue. It takes the bad protein and brings it to a blender. It recognizes a blender. You see the blender in the middle with the blue and pink and violet colors. And the protein, the bad protein, is inserted, is forced into the blender, and is now degraded into tiny small pieces that are called peptides. So now we got rid of this protein. So basically what we discovered this whole cascade where these proteins are, the damaged and the bad proteins are being degraded. Think about another disease, and now let's talk about the implications of what we discovered to diseases. You can see brains of patients that have Alzheimer's disease. You see on the right upper side, you see a normal brain, and look at the brain of the Alzheimer patients. You see that the brain of the patient is much shrunken. You see that you are losing the gray material, the material that dictates our high functions of intelligence, of memory, and so on. You can see also on the middle lower part of the slide, you can see again two brains. These are real brains that are kind of cut sections in the middle, and you can see on the right side, you can see the big distances between the different parts of the brain compared to the nice dense brain on the left side. So that's what happens in Alzheimer's disease. And the reason that in Alzheimer's disease, and not only in Alzheimer's disease, but basically in all diseases of the brain, in Parkinson's disease, in ALS, in different other types of dementia, there are aggregated proteins. Proteins are aggregated. They are somehow not recognized by the ubiquitin system. The ubiquitin system that we discovered look at them, but instead of removing them, it just doesn't remove them. They accumulate and they lead to cell death. These proteins, the aggregated proteins in the brain that should be normally removed, but somehow are not being removed by our system, are causing degeneration of the brain. And what you see is these spaces in the middle of the brain. Basically, you are seeing lost brain tissue. On the left upper side, I'm moving to another disease, very common disease, which is called cancer. You can see the liver in a CAT scan, and you can see in the liver a tumor. You can see on the left side, on the left upper side of the liver, you can see this bulging, and the bulging is a tumor. Now, tumors are being caused by proteins that are called oncoproteins. Proteins are proteins, and remember the oncos in Greece is a tumor. Remember the Department of Oncology in the hospital is treating cancer patients. So that's, you see here, so what happens in the body because of a mutation or because of a viral infection or any other reason, a normal protein is getting mutated, and mutated protein normally should be removed, but somehow the system that we discovered fails to see them, and they are driving the malignant process. And what you see is a tumor in the liver. So you already have an idea why it's very important to remove damaged proteins. We cannot leave them in the body. So you do need a system that was developed along evolution that will take damaged proteins and remove them. And if they are not being removed, and I can bring you numerous other examples, diseases are evolving. One group of big diseases, or very important diseases, are the neurodegenerative diseases, diseases that cause degeneration of the brain, Alzheimer, Parkinson, ALS, Huntington, and so on and so forth. And the other very big group of diseases are cancer. There are hundreds and hundreds of different cancers, and many of them are caused by proteins that used to be normal protein. They have undergone a mutation. A mutation was inserted into the structure of these proteins, and they, instead of being normal protein, they became oncogenic. They are leading to genesis, to the creation of tumors. And therefore, the system that we discovered is very, very important, not only to understand biology, but also to understand diseases. And as a result, people started to develop drugs. After we discovered the system, people started to develop drugs. And one of the drugs that you see here, you don't see a drug, you can see the bottle of the drug on the right side, on the right upper side. The drug is called Velcade. And the drug is fighting a malignancy of the blood. You know, malignancies of the blood are very common. In this case, it's a malignancy of the immune cells, of the B cells, and you see a typical patient here. You know, the experiment, these are relatively new drugs. The first drug came to the market in the 2000s, in the beginning of the 2000s, and you see that the experiment was done on this patient on October 1st, 2000. And what you see the galloping level of an immune globulin, of an antibody that is secreted by the malignant cells. So it's kind of a monoclonal expansion of cells of the immune system that secrete into the blood kind of a antibody that we can test and check. And by looking at the level of the antibody, we can tell what is the state of the disease. So you see that there is a galloping level of this molecule called IgA. Some treatment started and you can see a slight decrease, but then the disease relapses, it attacks again, and then started a treatment with a drug that is based on our system, with Velcade. It was invented and discovered after we came with our system, and it's an inhibitor of one of the components of the system, and you can see how the level of this protein goes down to almost zero, and actually they use the acronym CR. That's an acronym that is being used in oncology. It's called complete remission. The patient has basically not cured because the disease may relapse again, but currently the patient, after a year, you see that this is already October 1st, 2001, the patient is completely free from the disease. And look at the bone marrow of the patient on the left side. You can see that there is what we call a tumor burden. You can see that there are 41% of malignant cells. You can see the homogeneous view of the bone marrow. It's a biopsy that was taken from the bone marrow of the patient. The disease is a disease of the bone marrow where the malignant cells are occupying the entire space of the bone marrow and do not let any other normal cell to multiply, like red blood cells cannot multiply. So the patient is suffering from lack of oxygen because there is no red blood cell with hemoglobin to carry oxygen. The white blood cells cannot multiply. So the white blood cells protect us from infection. So you see that the patients are coming with repeated infections. They are bleeding because there are cells in the bone marrow that can prevent bleeding, that are involved in blood clotting, which are called thrombocytes. Basically, there is no blood to this patient. Everything is occupied by the malignant cell. And look on the right side, the bone marrow is normal, completely normal. You see different cells that are fulfilling different functions. And the tumor burden, the patient is not completely cured, went down from 41% on the left side to less than 1% on the right side. So the effectiveness of the disease was suppressed, of the drug, was that it could suppress the disease by 40-fold. So 97.5% of the blood came back to normal, and there are very few, 2.5% about of malignant cells. Look now what the drug can also do to solid tumors. What you can see here is a CT scan of a patient with another disease that is called NHL, or non-Hodgkin lymphoma. And what you see is a section in the chest. And you see the tumor. You see the tumor is 2.5 by 1.2 centimeter. You see the black is the air in the lungs. In the middle, you can see the big vessels and the heart. You can see the white of the ribs, and you can see the shoulder on the right side, the muscles of the shoulder. You can see the vertebra, the spinal canal, and on the right side, following one cycle of treatment, you can see that the tumor from this patient is basically gone. And it's again, if you do multiplication of the volume, about 90% of the tumor was gone. So you see that what is going on here, that when I started my journey in the late 70s, I had in mind an idea that I want to understand diseases, and it took about more than 40 years until the system matured, and now the drugs that are based on the system are saving the lives and improve the lives and the quality of life of millions of people. So in our lab now, we are developing a novel anti-cancer drug, and I hope that we are very close to clinical trials. And it's a very interesting one. What you could see in this slide, you can see the non-treated tumor and the treated tumor. These are all tumors that were taken from animals, from mice. So we inject the mice with, with 1 million cells of a tumor, of malignant cells, and they develop into a tumor. But the mice that are treated, you can see that the tumors are very small. What we are doing, actually, I want to explain. So what we are doing, it's kind of a trick. First of all, it's a little bit, it's based on our system. And I'll give you an explanation. I first want to give you an example, a metaphor, and then go to the drug that we are developing. It's a very tricky drug, but a very, very, very smart one. Let's go to something completely else. Let's go to thirsty. When you feel thirsty. When you feel thirsty, you are drinking, right? There is a center of thirst in the brain that tells you that you are thirsty. So you drink, but you have to drink quite a volume. You have to drink like a can of Coke in order to feel full. If you take just one sip, it will not give you a feeling of fullness. Why you have to do it? Because there is a center in the brain that sensing the volume of the concentration of the ions, the volume of the drink. It stretches the stomach. So you get a lot of information of how much you drank, and then this brain center tells you, okay, you don't need to drink anymore. Everything is fine. If I take an electrode, now I'm still in the thirst, forget about cancer. If you take an electrode and you put it in the brain of the mouse and you stimulate the thirst center, the mice will die of thirst. He will not drink at all. He will look at the water. You give the mouse as much water as he wants. The mouse will always, because of the stimulation, the center will tell the mouse, you don't have to drink. It's like you are drinking a can of Coke, but you have another 10 cans are sitting in front of you on the table, and you will not touch them because you don't need them. So you can deceive the body by stimulating a center that will tell the mouse, don't drink. You're okay. Everything is fine. What we did the same for the cancer cells. Cancer cells are much more hungry than normal cells. They eat a lot. So we found a compound that is hitting the satiety center of the cancer cell. And we give this molecule. So the cell says, I don't need to digest proteins. I don't need to eat. Everything is fine. And the cell are committing suicide. They are going through a process that actually was discovered by British scientists. It got the Nobel Prize, Sidney Brenner and John Salston, and one American. They got the Nobel Prize in the early 2000s. And we are basically deceiving the cancer cell to think that everything is fine. And it doesn't need to take proteins from the outside and to digest them. And the cell basically commits suicide. So you see that it took quite a long time. I started my career as a surgeon, and I ended up, I changed my route along my career in the middle, and I decided to become a scientist. When I became a scientist, I didn't know where the route is going to take me. We discovered, we asked a question of how proteins are degraded, how they are destroyed in the cell. At that time, we had no clue that it will be involved in disease mechanisms and in drug development. But now, I can look back with some pride, I would say. I'm still, you see behind Cambridge, behind my photo, but actually I'm sitting in my laboratory. I have a very active laboratory, and we are very active in science. and we are developing the drug, and we are doing other things. But I think that the main satisfaction is that I could help patients in a very big way. Because as a doctor, when I was operating, you could operate once a day, twice a day, depends on the length of the operation. And then the next day you don't operate because you have to go to see the patient that you operated the day before. And it's very nice, and you talk to families, you feel that you help people. But if you are lucky enough in science, you can impact the lives of millions of people that you don't know where they are, you don't know what their name is, but nevertheless, you know that last year in the world, there were millions of people with these blood diseases, and they were all benefited from the drug that was developed by you. Let me end up with something which is very interesting because people don't know about it, about the Nobel Prize. I don't. Let me just go to full screen for 1 minute to the Nobel medal. Okay, do you see it?
We can say it all good. Thank you.
Okay.
We
So
can say it.
the Nobel medal, you see, this is my personal medal. You see on the right side, my name is inscribed on the bottom, Aaron Ciechanover, and the year is 2004. So it's 20 years now. But what is nice, on the front side, you see the face of Alfred Nobel, and it says that he was born in 1833, and he died in 1896, and he, ordered, asked in his will that all his property will go to distribution of 5 prizes, not 6. Now there are 6 in medicine and in chemistry and physiology, in literature, and in peace. But the back part of the medal is very nice. Actually, the real medal is sitting on my desk. I don't know if you see. Can you see me? I'm holding it now in my hands.
Can you lift it up, please?
I'm holding it. But anyway, there are 2 women. And you
If
see
you can slightly lift it up, we'll be able to see, I think.
What did you say?
Can you lift it up to the camera?
Yeah.
Okay.
Here it is.
Thank you. Wow, we can see it. Thank you.
Okay, but I want to show you I'm not showing it to you because I'm proud of it. I'm showing it to you because this will give you an idea about what's going on in science and in nature. You see the right woman, her name is Scientia, science. You see, it's written science on the right side. The left woman is nature. Now, nature is holding its secrets in a horn, in a cornupia, very close to her chest. Science is lifting the veil from the face of nature and exposes the horn, the cornupia, with the secrets of nature. So this is the interrelationship between science and nature. The role of science is to uncover the secrets of nature in order to benefit people. So this is the story of the medal, which I think is kind of very nice. You see also the diploma. This is my diploma, and it says that it's in Swedish, that the Royal Swedish Academy of Sciences decided to give the Nobel Prize to me on December 10, 2004 for the discovery of the ubiquitin system. So I think that this is the story. We are close to the end. I left less time than I thought that I am going to leave for questions, but if you have questions, I will be more than happy to answer them.
Thank you so much for your excellent presentation. We really enjoyed it. Thank you so much. Let me redirect some questions to you. Any questions in the live audience? Yes, please. Thank you. The question is, can you hear me?
Yeah.
The question is, how did you find ubiquitin in specific? Was it an accident or were you directing experiments towards it?
Well, it's a very good question. Everything started with hypothesis. We knew nothing about ubiquitin, and we didn't mean to discover ubiquitin. we asked a biological question, and what we meant is to discover a system that degrades proteins. So we put a question. Now, once you have a question, of course, you need to design a biological, not to design, but to identify a biological question, a system that will give you an answer. You can ask many questions. You can ask, okay, I want to live 250 years. That's a very nice and legitimate question. But if you don't have a system in order to study aging, then, the question will remain up in the air. So we said we hypothesized that there must be a quality control system that is removing damaged proteins. And we wanted to discover it. So that's the advantage of having two types of education. And this is the point that I wanted to highlight for you, and I forgot, but it's good that the question brought it back. Remember, I'm a physician, and now I'm also a biologist. So I think that commanding the two languages is extremely helpful. So my mentor, Professor Hershko, is also a physician. So he said, where can we find a biological question? So we said, we can find it in the red blood cell. In the young red blood cell, which is in the bone marrow, and it's not injected yet, it's not circulating yet, is degrading all its system. It's getting mature. It degrades the nucleus, it degrades the mitochondria, it degrades the respiratory system, the protein synthesis. At the end of the day, the red blood cell that remains after all these proteolytic processes is a very primitive cell. It contains hemoglobin, of course, the red blood protein, and it contains some enzymes that are making energy out of glucose and some other few proteins. But about 90% of the systems are degraded. Why? Because the cell doesn't need them anymore. So we said, wow, here we have a cell that doesn't need its system, and it degrades them. So maybe the system that degrades these proteins during the maturation of the red blood cell will be the system that we are looking. And that turned out indeed to be the case. So what we did, we took the young red blood cells from the bone marrow. I will not go into techniques how we did it. And we opened the cells, and we started to isolate the proteins until we discovered we put an assay in what we call an in vitro assay, a test tube assay, to degrade proteins. And we started to separate all the components and to put them back like a hand watch. If you want to know how a hand watch is working, what you do, you take a screwdriver and a cloth, you take your hand watch, you take away you disintegrate it, you dismantle it to all the pieces, and then if you put all the pieces back and it works, you know how the watch is working. That's what we did with the red blood cells. We took the red blood cells, we took the juice of the red blood cell, the cytosol, and we disintegrated it, and we started to put it together until we discovered the different component, and ubiquitin was one of them. But at the end of the day, we were limited by our technique. We were working at the late 70s. When the real system was discovered in the early 2000s by exposure of the human genome, it turned out that the ubiquitin system that we discovered contained more than 2,000 proteins. About almost 10% of the human genome is the system that we discovered. We didn't know how big it is. So think about how important is garbaging in the eyes of the cell and in the eyes of evolution. 2,000 proteins are involved in garbage collecting and disposal of the cell. That's how important it is. Almost 10% of the entire human genome is dealing every day with the garbage collection and destruction. But the beginning was having an idea that there is a system, finding a cell that we knew a priori that might have it, and that was the maturing red blood cell, and then going to the bench and starting to take the cell and to see whether such a system is indeed there. And it took me 5 years of a PhD and working in the cold room and separating all the components and putting them together. What I wanted to tell you also, that after the 5 years, I decided to fly out of the nest, because you want to build yourself as an independent scientist. And I actually weighed, I must tell you that I weighed between the UK and the USA. Oxford was on the on the table, but then I decided to go to Oxford of the East Coast of America called MIT, and I spent 5 years at MIT expanding my biological education, and then I returned back. So this was also part of the education. I wanted to highlight to you, because you are young students, the fact that I decided to acquire a second language. And being polyglottal is a big advantage in the modern world. And being a scientist and a physician has a huge advantage, because I can see the entire distance between the sick patient, and actually I'm in a medical school, and the hospital is nearby. Actually, it's 50 meters from me. And we all the time work on disease models and a biology, so I can combine the two. And as a matter of fact, now people are living long, and you have enough time to develop this dual career. The medical schools in both Cambridge and Oxford have this pathway for dual education, and if you're interested in both medicine and science, it's highly recommended. So anyway, I'm sorry for very long answer for a short question. Yes, go ahead, another one.
Thank you so much. I think you answered my second question as well, which was some advice to the young students we have who are deciding their career, subject, and colleges. So thank you so much for the beautiful answer. Let me check if we have one more question from the audience. Okay, we don't have anything at the hall, and the Zoom seems fine. Do you have any final words for our students here at CCIR?
All I can tell you is that the world is ahead of you, and science is so much interesting. And I'm not trying to convince you to go to biology or to medicine. I think that you should go to whatever you feel that you are good at. If you feel that you want to be a dancer and you are good in dancing, go and be a dancer. If you want to be a designer, you feel that you want to be an architect, go to be an architect. But the only condition for success, I think, is that you are really good at something, that it's coming out of your fingers. You do understand it. You see it in all the full 3 dimensions. And I decided that medicine is not for me because I wanted to know more. I didn't feel comfortable, and I was not hesitating for a minute to take studies of 7 years plus, residency in medicine. To put it aside, I'm still using it. I speak the language, and to go to something that I feel more comfortable with, that it's flowing in my blood. And the idea is not to get the Nobel Prize. The Nobel Prize came much, much later. The idea is to enjoy life. And I can tell you that I've been to science for more than 50 years. I'm now in my laboratory. Every day is a new day. We are doing today what we didn't do yesterday, and we did yesterday what we didn't do the day before. We are always, renovating, because in science you are doing something that nobody has done before, right? Otherwise, we are not in a school. We are in a university. We are in research. In research, you are discovering. You are not transmitting already known knowledge. And I think that it's fun, and I was lucky enough to impact the lives of millions of people worldwide, but that's not the aim. The aim is really to feel good about what you are doing, to contribute to society, and basically to enjoy life. The word enjoyment is, has many meanings. I don't think that in life you have a purpose. People are, accumulating billions of dollars, and then they die and they leave everything behind. I think that the idea is to move in life from one enjoyable experience to another enjoyable experience, and just, have fun. So I wish you all the very best in your future career.